This is part 2 of our look at color perception, and we're really going to get into the heart of it, looking at theories of sensory coding. We can start off by trying to narrow things down a little bit. For our scotopic vision, that is, vision under low light conditions, only our rods are functioning. We do not get a full color experience. This allows us to conclude that our cones are responsible for our color vision, but that really doesn't narrow it down much. How exactly do our cones allow us to perceive colors? I'm going to go chronologically. I want to talk about the first theory of sensory coding, put together by two different individuals. They did not collaborate, so this is put together over many decades. Thomas Young, in 1802, thought about all the colors that we can experience. He had no idea how many there are, if there was 10 million or 2 million, or exactly what number it was, but he felt it was a pretty large number. And he thought it was pretty improbable that we would have a separate type of receptor for every different shade that we can perceive. That seemed really implausible to him. Instead, he proposed maybe we have only a limited number of receptors, like, say, 3, and these 3 work together to allow us to experience the entire range of colors. Each one of these hypothetical receptors would be sensitive to a different wavelength or a range of wavelengths. That's pretty forward thinking in 1802. It took quite a few years to get other missing parts to this. So the next part is attributed to Hermann von Helmholtz, but really he followed in the work of James Clerk Maxwell, so I have to give him credit. It was von Helmholtz who provided evidence that, yes, there are actually 3 primary colors. We can put these together according to the definition of primary color so that we can match any given color. And again, this was determined experimentally by taking color photographs. You can reproduce the entire color palette of the world around you using only 3 primaries. If you use 2, you're not going to capture all the colors. You could do it with 4 or potentially 1,000, but that's kind of overkill. Okay, that's still the theoretical side of things. This is the heart of the evidence supporting the Young-Helmholtz trichromatic or 3-color theory. You can do color matching experiments. So give people a color and then give them 3 primaries. Ask them to adjust the 3 primaries in different proportions until they can match the given color, and people can do that. You can do that with 3 primaries. You cannot do that necessarily with 2. Even stronger evidence comes from physiology. You can measure the wavelengths of light that are absorbed by the cone receptors. This is done using a microspectrophotometer. That was not possible in the 19th century. That did take a few more decades before that was possible. And it was discovered that cones come in 3 types. Each different type contains a different pigment. One type is called erythrolabe, which means literally red catching. This one most strongly absorbs long wavelengths. Its peak is about 558 nanometers. That's not a red color, though. Red is about 700, 720, so we have to be careful about not taking this literally. There is chlorolabe. If you think of chlorophyll, these are the green catching, and then cyanolabe, which is literally the blue catching, which absorbs shorter wavelengths. Different wavelengths will produce differing responses from each receptor type. What does that mean? Okay, let's imagine we're shining a light of 550 nanometers into the eye. So that's right there. It's a slightly yellowish green. This is going to barely activate the blue cones. They're barely sensitive to that at all. The so-called red cones will have a much stronger response, and the so-called green cones will have the strongest response of all. Taking a look at that pattern of activity, the brain interprets it as, oh, this is 550 nanometers. This is some pretty solid evidence, This looks like this is the way to go. Trichromatic theory is the way to go. It is a form of specificity theory of sensory coding, if you're keeping track. But there is also another theory called opponent process theory, proposed by Ewald Hering. Hering's approach was partly based on thought experiments and people's experience, and the fancy term for that is phenomenological observations. Asking people questions like, how many primary or basic colors do there seem to be? Well, red is a pretty basic kind of a color. Green, also kind of a basic sort of primary color. Blue as well, and yellow. So there we go. There's 4 primary colors. Wait, 4? Yeah, 4. Red, green, blue, and yellow. Now, nobody's sitting around arguing for brown or chartreuse or teal. Those don't seem to be kind of fundamental basic colors. Red, green, blue, and yellow. By my count, that's not 3, that's 4. Also asked questions about combining colors. What happens if you combine certain pairs of colors? Does anything weird ever happen? Like, I don't know, complementary colors that you combine them together and the color cancels out and disappears? Whoa, what's going on there? How do you explain that, trichromatic theory? What's that? Yep, crickets chirping. Yeah, that's what I hear. Hering proposed that colors are basic and they come in opposing pairs, hence the term opponent process theory. And this is the complementary colors. So there is red, and its kind of opposite is green. There's kind of a bluish, and its opposite is kind of a yellowish, orangish. And of course, there's black and white as well. As intuitively appealing as this is, it's not exactly solid, hard evidence. Let's try and uncover some harder evidence. What I'd like you to do, after you kind of look around this for a second, is focus your eyes on this black spot, okay? Do not move your eyes around. It is important that you just stare at that black spot. You are allowed to blink. I don't want your eyeballs to dry out. That would be gross. Okay, so you might be able to direct your attention around a little bit without moving your eyes, and you might determine that you're kind of looking at a bluish castle, kind of a yellowish, orangish background or sky, which is pretty weird. But okay, just keep staring. Just keep staring at that dot. It'll just take about 30 minutes. You might want to pause the video. No, I'm just kidding, for Pete's sakes. Okay, this should work. Now what I want you to do is to keep looking at this dot as I flip the slide, and now you see it in full color until you move your eyes. Wait, where did the color go? Okay, look back at the dot. Some of the color is back. Move your eyes around again. Color is gone. Look at the dot. Color should still be there. Blue sky, kind of a grayish castle, green grass, and that will eventually fade and disappear. What the heck happened? Where did those colors come from? So clearly, this is a black and white photograph, and the first photograph was the wrong colors. All right. This is some pretty strong evidence. Trichromatic theory cannot account for this, but opponent process can. What hopefully you experienced was an after image. What you experienced was the opposite of what you were staring at initially. So the colors were all wrong. So when you were staring at that first image, the sky was not blue. It was kind of the opposite, kind of a bit of an orangey yellow. But then afterwards, it did turn blue. Now you can create one of these. It's called the Spanish Castle. You can create one of these yourself with any photograph you have. All you have to do is load it into some paint software. The first thing you have to do is go to the color menu and go to negative. So turn it into the negative image and save that. Reload the original. Go to the color menu again, change it to grayscale, and save that. And you can do exactly the same thing as you experienced here. Again, trichromatic theory does not make any predictions about why you should see the opposite of the color that you were staring at during that adapting phase. Here's some more evidence. Okay, so kind of a cool psychedelic swirly pattern. All right, so there are some swirls, and if you take a look at them, there's kind of I guess this is a kind of a light green, kind of a lime green swirl, and then the other swirl is, well, it's kind of a bluish green kind of swirl. All right, and then there's some other kind of narrower stripes of blue and yellow you can see. Let me just take some of those away and then see what happens. Okay, so the yellow is gone. Okay, but the swirls, there's still like a lighter green swirl, and then this is kind of like a darker green swirl now. Let me take away the blue and see what happens. Wait, what? Yeah, the swirls are actually the same shade of green. Adding the blue and the yellow made you misinterpret or misperceive those colors. Trichromatic theory cannot explain this either. The colors seem to be interacting with each other. If you think you see colors in an absolute sense, this shows you that no, you don't. You see them relative to each other. Crazy. All right, so that's another piece of evidence. This is called simultaneous color contrast. It doesn't require an adapting phase. You don't have to stare at a stimulus for like 2 minutes or anything. It just happens immediately. And it's basically if you surround one color with another color, that changes the appearance of the color. Again, trichromatic theory cannot explain this, but opponent process, it can. We also have a third line of evidence based on habituation. This works really well with infants. So one of the criticisms of this approach is, well, it's all about how adults use language. We have words for red, green, blue, and yellow, and we've been contaminated by that language. And that produces the responses that Haring has found. But how can we separate language out? How can we go to people who don't have language? Babies. Okay, babies have not yet learned language, but how are they going to communicate? Fortunately, we can take advantage of the fact that an infant gets bored pretty quickly if they look at the same thing. So here's a very clever experiment from 1976. The first thing they did was present 510 nanometer light. So it's kind of a dark green here, you can see. And they presented that to 4-month-olds until they got bored. It doesn't take very long. They're like, oh, it's that color, and I'm bored of it now. What else you got? Okay, so after presenting this 510 nanometer light, they presented one of two other lights. This is 540 nanometer light, and the infants were like, that's still boring. Or they changed it from 510 to 480. And now the infants were like, oh, I love that. That's so interesting. Of course, they weren't talking. You do understand that, right? They're infants. They're 4-month-olds. Okay. Now, unless you have some form of color blindness, you will experience the same thing. The 540, it's just like another shade of green, but 480, well, that's blue now, isn't it? Now, notice the amount of difference. The amount is 30 nanometers of difference. We're either going up 30 from 510 or going down 30. But if we go down, we cross a boundary from green into blue. So what we get from these infants in their pattern of looking, of being bored or excited, matches what adults tell us verbally using words. Yeah, 540 is still a green, but 480, it's different. It's a blue. What this evidence tells us is that babies who haven't yet been contaminated by language categorize colors very much like adults do. And this is just one piece of a line of research that showed that there are 4 basic color categories. Nice. But wait, we still have more evidence. Again, opponent process theory really had to kind of fight against the tide of trichromatic theory, which was very intuitive and appealing and had its own evidence to back it up. It's really hard to argue against physiological evidence. And there is physiological evidence. We can go to center surround cells. You thought you didn't have to remember any of that. No, I'm bringing it back. Center surround receptive fields are basically there to detect contrast. contrast between the presence of light and the absence of light. But what about color? Are there any center-surround receptive fields that respond differentially to color? And the answer is yes. We can go back to 1958. This is a study of fish, and specifically horizontal cells of fish responding to different colored stimuli. So it shows actually the output of two different cells. So one is on the left, one is on the right. The cell on the left goes down below its baseline rate of firing when it's presented with basically bluish and slightly greenish colors, and it increases when it's more yellowish colors. Well, blue versus yellow is one of the opposing pairs proposed by Herring. Another kind of cell is on the right. This one turns out to be inhibited by green and more excited by red. Again, also predicted by Herring, green versus red. There aren't any cells that are blue versus green or yellow versus red. So this is an opposing response to colored pairs. Now this leaves us in a weird position of evidence supporting trichromatic theory and evidence supporting opponent process theory. How can we reconcile these two? They're both correct, but they can't be completely correct without the other one. That still took a few more decades, and a proposed neural wiring diagram for all of the cells that are involved. And this is called two-stage theory, proposing at a very early stage of visual processing two different stages when it comes to processing wavelength and color. The first stage occurs at the level of the photoreceptors, the cones. So I've drawn two cones here, two different types. The green one with M for green and L for red? What? Okay, I don't want to call them green cones and red cones. That's a bit of a misnomer. So instead I'm going to use M for medium wavelength, because green is right in the middle of the spectrum, and L for longer wavelength. Okay, so we've got basically green detectors and red detectors, and these are wired up in opposition to each other. If there is green light that strikes this part of the retina, it will bounce off of this long wavelength cone, basically be ignored. It'll be absorbed by the medium wavelength cone, which will then release excitatory neurotransmitter to this big cell. If we have longer wavelengths, it's going to excite this cone, which will in turn release inhibitory neurotransmitter. This big cell down here is a neuron, and this neuron is excited by green, G+, and inhibited by red, R-. It's showing color opponent pairs and how they might be wired up. So, up here at the level of the photoreceptors at the cones, we have some evidence that it fits in with trichromatic theory. It doesn't contradict it. And then down here, where we get this excitation and inhibition, this now conforms to opponent process theory. This is one possibility. There's another possibility in which the medium wavelength cones release inhibitory neurotransmitter and the long wavelength cones release excitatory, and we'd have a G- R+ neuron. I didn't want to draw every single possibility. Okay, so now we have part of what we need. We have a proposal for how we can wrap together trichromatic theory and opponent process theory, and we've got some color pairs, green versus red, essentially. Now, really, all I have to do is show you the same thing for blue versus yellow, and then we'll be done and we can all go home. But the problem is there are no yellow cones. Okay, there are only three types of cones: short, medium, and long wavelength. So how do we account for the blue versus yellow? It's a little bit more complex. Now I'll start off with the less complex part, the short wavelength cones, which do detect shorter wavelengths, the blue end of the spectrum, and when we have blue light striking this cone, it will release excitatory neurotransmitter and it will excite this neuron. But we don't have yellow cones. But we do have is metamers. So if I shine yellow light onto this part of the retina here, the yellow is going to have no effect on the short wavelength cones at all. Yellow light, however, is going to be, to some extent, absorbed by the medium wavelength cones and by the long wavelength cones. This is why I don't really want to call them green, blue, and red. I want to call them short, medium, and long. So the color yellow in the spectrum is kind of between green and red. So yellow light activates the M cone and the L cone. Now both of these are going to be releasing excitatory neurotransmitter, and they're going to activate this intermediate cell. This cell, when it's activated, will release inhibitory neurotransmitter. I think this part of it is a bit difficult to kind of wrap your head around. The presence of yellow light is going to cause a decrease in the firing of this color-sensitive neuron. So it's activated by blue, B+, and inhibited by yellow, Y-. And yes, we can also have the kind of opposite. We can have a B- yellow plus. I didn't want to draw every single different version of it. Now, exploring this at another level of abstraction, we can say that what's happening here is there's a difference function being calculated. Red versus green, blue versus yellow. This is a really neat way to reconcile trichromatic theory. We haven't violated that. All of this is based on only 3 cone types. It also includes opponent process theory because it proposes at the neural level opposing processes of excitation and inhibition. We can continue to look at how the visual system processes color by going deeper, going into the cortex. We can go to area V1 in layers 2 and 3. In the blobs, we find color opponent cells. They have a center surround organization, but now it's all about color. It's not about presence or absence of light. So one example here is a red on center and a green off surround. So it's looking for the difference between red and green. Again, this is just one possibility. We also have red minus center and green plus surround. Again, I just didn't want to draw every single possibility. These cells aren't just looking for contrast between presence or absence of light, but presence or absence of green versus red. And no, we don't have any red versus yellow cells. It's all green versus red. Still sticking with area V1, but also going to area V4, there are also some cells that have even more complex color processing. They have double color opponent cells. What? Yeah. So what happens if we shine red light into the center? It will excite the neuron. Green light will inhibit it, while at the same time, red light shined on the donut will inhibit the neuron, and green light will excite it. Again, it's all about red versus green. There's no red versus yellow or green versus blue. So even as we go deeper into the cortex, we can see this opponent color organization is still preserved. We can learn a lot from color deficiency as well. Notice I'm not calling it color blindness. That term is not preferred for lots of reasons. If you ever go for an eye test, you can be checked for color deficiency. It's a test called the Ishihara test. And it's got these kind of dots and blobs in there. And if you look at that, you should be able to see that there's a number in there in a different color. And that number is 14. Okay, I know, it's a dad joke. It's 29. It's 29. You should be able to see 29. I've got some more for you. You see the number in there. The answer is 12. Here's the next one. Can you see it? You might have to squint your eyes a little bit. This is a tricky one. The number is a 6. And here's another one. What number do you see? Are you just Guessing? Is it hard to see? Are you really worried now that you have color blindness? Okay, this is called a hidden digit plate. If you have red-green color deficiency, you will see the number 2. If you don't have this deficiency, you'll just see a bunch of colored circles. So I tricked you a little bit, didn't I? Okay, one more. Do you see a number? The number is 42. That's a good one to end with. Okay, so those are part of the Ishihara test. So what do you do if you have some form of color deficiency? Is there any corrective surgery? No. Are there special glasses you can buy that... No, you can't. I know there are lots of claims made on the internet about these special glasses that let people see colors for the first time. Now the answer is it will allow them to better discriminate certain colors, but it's actually going to impair their ability to perceive other colors. No, there are no magic glasses yet that can compensate for color deficiency. I've talked about color deficiency before. I talked about rod monochromacy. So these are people who have no cones at all. The incidence of that is 1 per 100,000. So pretty rare, but not so rare that we have never heard of it. There is also cone monochromacy. These people have only one type of cone. Now they do have rods, but importantly only one type of cone, not 3. And if you kind of think it through logically, there are 3 subtypes of cone monochromacy, but I won't go through them all. I'll just leave it at the fact that blue cone monochromats are more common for some reason. There is also dichromacy, in which people have only 2 types of cone. Again, there are 3 different subtypes, and I will go through these. Protanopia means that people are deficient in their long wavelength cone. They will perceive the rainbow spectrum of colors as ranging from blue to yellow. People who have this have a great deal of difficulty distinguishing red and green. In deuteranopia, people are lacking the medium wavelength cone. They will perceive the spectrum as blue to yellow? What? And they also have difficulty distinguishing red and green. Wait a minute, did I get this mixed up? That sounds like protanopia. No, I didn't get it mixed up. It's not exactly what you might expect based on kind of a simple conception based on trichromatic theory. The third one is tritanopia, in which people are missing the short wavelength cone. This is actually very, very rare. They perceive the spectrum of colors as ranging from turquoise to red. Your textbook says blue to red. I dispute that a little bit, and there is some other evidence that backs me up in this. People with tritanopia have difficulty perceiving blue and yellow. So just to give you some sense of what these different forms of dichromacy are like, here are some visual examples. Okay, so at the top, the normal spectrum, so ranging from red, orange, yellow, green, blue, violet, and then here's what a person with protanopia would experience. So that's pretty much seeing blue to yellow, and that's it. Deuteranopia also technically blue to yellow, but kind of different shades of yellow in there. And then tritanopia from blue, no, to me again, that looks like turquoise. There's a bit of green in there, to red. There's also websites you can go to and plug in any image, and they will simulate many different forms of color deficiency. So there is the original image. This is what a person with protanopia would experience. Okay, so the green has been kind of sucked out of the leaves. Pretty much the same thing for deuteranopia. And there's tritanopia, in which the blue has been kind of sucked out of it. And as a result, the flower bud looks much more red. Now, there are other kinds of color deficiency as well. There's something called anomalous trichromacy, in which people aren't missing anything. They've got all the equipment in their eye, but for some reason, they're not so good at discriminating between hues. It might be because the cone type is slightly abnormal, possibly due to a genetic defect. Kind of a general thing is that males are more affected by color deficiency than females, and that of course indicates it's sex-linked. that a lot of the genes for producing photopigments are on the X chromosome.